This protocol demonstrates how to image dynamic events in early C. elegans embryos with Normarski DIC optics or fluorescence by making use of widely available microscopes and open source software. The protocol begins with describing how to dissect C. elegans embryos and mount them for imaging. Then, proper setup of the DIC Normarski optics is reviewed to ensure optimal images at the highest detail. Calibration of wide-field epifluorescent illumination is also reviewed for collecting good fluorescent image data. Teamed with open-source software, good microscopy can visualize cellular dynamics in a developing embryo.
Visual demonstrations of this method is critical, as setting up DIC optics properly is hard, since the terminology can be confusing and the proper technique is hard to describe in written form. In addition, researchers should be careful to minimize light exposure when performing fluorescence imaging in order to limit phototoxicity and photobleaching.
To prepare embryos for imaging, first make two mounting solutions. One is melted Vaseline and the other is 2 to 3% agarose in buffer. Denser agarose can be made if the structures to be imaged are very fragile. Being careful to avoid boiling, both stock solutions are melted in the microwave and then stored as 4 to 5 milliliter aliquots. On the day of imaging, melt an aliquot of Vaseline solution in a 65 to 70 degrees Celsius heat block and melt 1 aliquot of agarose solution in the microwave, again avoiding boiling. Then, to keep these solutions molten, store them in a heat block. Next, cover the top and bottom of 2 slides with a strip of lab tape and position a clean slide between the